Welcome to this Carbon Academy tutorial on AS level biology. In this video we'll be looking at membrane transport and transport proteins. This is part 2 of the cell membranes topic. Here are the aims of this tutorial. We'll define and describe the types of passive transport, list and explain the factors that affect passive transport, then look at defining active transport and explain the factors that affect it, and finally look at other mechanisms by which membrane transport occurs. So let's get started. There are essentially three main types of transport. One is passive transport, which consists of simple diffusion, facilitated diffusion, and osmosis. Another one is active transport. And finally, we have endocytosis and exo-cytosis, which are other mechanisms of cell transport. So let's first look at passive transport. So passive transport is a net movement of substances from an area of high concentration to an area of low concentration down a concentration gradient. In passive transport, you do not require any energy. In simple diffusion, there is diffusion through the phospholipid bilayer. In facilitated diffusion, diffusion occurs using channel or carrier proteins. And in osmosis, there is diffusion of water through a partially permeable membrane. Let's first look at how the membrane proteins function. Firstly, the channel proteins are only involved in facilitated diffusion. And channel proteins can either be gated or always open. Examples of channel proteins that are always open are aquaporins, which allow water to be absorbed more in kidney tubules. Another type, like I mentioned, was that they can be gated or always open. Gated channel protein examples include voltage-gated sodium ion channels and ligand-gated acetylcholine ion channels. Binding of acetylcholine opens up the channel protein to allow sodium through. This will be discussed further in my A-level videos. Now let's look at how carrier proteins function. Carrier proteins are involved in both active transport and facilitated diffusion. When a molecule binds, the carrier protein changes shape, and this lets the molecule into the cell. Examples of carrier proteins include GLUT4, which is a carrier protein involved in the facilitated diffusion of glucose into adipose cells or fat cells, and also sodium-potassium ATPase, which are carrier proteins involved in the active transport of sodium ions out of the cell and 2 potassium ions into the cell. Now let's look at osmosis. You should be familiar with this from GCSE. In osmosis, there is net movement of water from an area of high water potential to an area of low water potential through a partially permeable membrane. But what is water potential? Well, water potential is the concentration of the water molecules. And so the greater the concentration of water, the higher the water potential. The highest water potential you can have is 0 kilopascals, kPa for short. And this is where the water is pure and there is no solutes present. Presence of solutes lower the water potential, making it more negative. As this diagram indicates, this symbol, the psi sign, is the symbol for water potential, and minus 50 kilopascals is greater than minus 200 kilopascals. So therefore water will move from this region to the region to the right through this partially permeable membrane. Remember, the solutes cannot pass through this membrane where water can, and once equilibrium is reached, no more osmosis occurs. Now let's look at what is tonicity. Again, this is something you would be familiar from GCSE. Tonicity refers to the concentration of solutes outside the cell. Remember, water always moves in the direction of high solute concentration. There are three types of tonicity. Firstly, we have isotonic, where the two solutions, i.e. the concentration of solutes inside the cell is the same as the concentration outside the cell. Then we have hypotonic, where the water potential inside the cell is lower than the outside, essentially saying that the concentration of solutes outside the cell is less than the concentration of solutes inside the cell. And hypertonic refers to a lower water potential outside the cell than inside. Hypertonic is the same as saying there's a high concentration of solutes outside the cell than inside the cell. Hope that made sense. Now let's look at the factors affecting the rate of diffusion and osmosis. Firstly, we have surface area. Increase in surface area increases the rate of diffusion, which again you should be familiar with from GCSE. Then we have the concentration or diffusion gradient, same term really, and water potential gradient. And a steeper gradient increases the rate of diffusion. I like to think of this as a slide in a park. The steeper the slide, the faster you go down. So the steeper the gradient, the faster the rate of diffusion. Then we can look at the length of diffusion pathway. If the diffusion pathway is thin, then there's going to be a higher rate of diffusion. If it's long, it's going to take longer. And how could we not forget temperature? Temperature pretty much affects everything, and it also affects diffusion. If there is a higher temperature, the molecules have more kinetic energy, so they move faster, and this results in an increased rate of diffusion. Finally, we have the size of the molecules. Small molecules diffuse faster than bigger ones. Why? This is because small molecules weigh less than the larger molecules, so you do not require as much energy to move the small molecules. So they essentially move faster for the same energy. Now let's look at factors that affect facilitated diffusion. Factors that affect simple diffusion also affects facilitated diffusion, but additionally there's one more. Facilitated diffusion is limited by the number of channel or carrier proteins available in the cell membrane. Therefore, if all these proteins are occupied, no further facilitated diffusion occurs. It's similar to an enzyme substrate complex curve. If all the enzyme's active sites are occupied, no further ES complexes can be made. Now let's move to active transport. Active transport is the movement of substances against a concentration gradient. So it's like climbing up the slide. And this requires energy in the form of ATP. Active transport only uses carrier proteins. No channel proteins are involved in active transport. And there are many types of carrier proteins. Firstly, we have uniport proteins, which only lets one molecule through. And we have co-transporter proteins, which let two molecules through. There are two types of co-transport proteins. One is a symport, which allows two molecules to move in the same direction. And another one is an antiport, which allows two molecules to move in the opposite direction. Let's look at factors that affect active transport. Firstly, we have the number of carrier proteins. Again, greater the number of carrier proteins, faster the rate of active transport. Then we have the rate of respiration. Higher the rate of respiration, faster active transport. If there is no respiration occurring, there can be no active transport. So it makes sense that the cells that carry out active transport need a lot of mitochondria. Then we have the number of ATP available. Again, more mitochondria means more ATP means more active transport. Also the speed of carrier proteins. Carrier proteins work at different speeds, and faster they work, faster the rate of active transport. Finally, let's look at endocytosis and exo-cytosis. So endo means in, cytosis refers to the cell, and so endocytosis usually occurs when a cell ingests a large molecule or large substances. And usually this is large cells or macromolecules like starch. In this scenario, we will take the example of bacteria. You will be familiar with phagocytosis from the immunity topic, and phagocytosis is a type of endocytosis. Exo means out and cytosis refers to cell again, so exocytosis occurs when cells secrete substances into the blood or to a junction. Usually, cells secrete hormones into the blood and neurotransmitter into junctions called synapses. Note that endocytosis and exocytosis are active processes, so they require energy in the form of ATP. Endo and exocytosis allow mass transport. So here are the aims of this tutorial. As usual, use them as questions to consolidate your learning. I thank you for watching, and I'll see you in the next one.